During early interphase, nothing out of the ordinary is happening. The cell is performing its function, and the diploid number of chromosomes are present in the nucleus. However, as the cell transitions into late interphase, everything in the cell duplicates. Inside of the nucleus, each chromosome duplicates itself, forming identical copies of the chromosomes called sister chromatids. Outside of the nucleus, the centrosomes and the spindle fibers duplicate themselves. All of the organelles present in the cell are also duplicated, but for the sake of showing meiosis, these organelles will not be important. After the chromosomes duplicate in late interphase, the nuclear membrane disintegrates and the sister chromatids find their homologous pairs, forming a synapse and creating a 4-part chromosome structure called a tetrad. Inside of the tetrads, the sister chromatids and the non-sister chromatids are so close to one another that they exchange genetic information during a process called crossing over. Sister chromatids and non-sister chromatids will exchange their DNA for alleles, creating new combinations of alleles on a chromosome. Crossing over increases the genetic variation of the gametes, thus creating more diverse offspring.
Over the course of metaphase 1, the newly made homologous pairs begin to line up. The tetrads move towards the middle of the cell and form a line towards the equator. The centrioles and spindle fibers move to opposite sides of each other. The cell is 2N, or a diploid cell.
At the beginning of anaphase 1, the homologous chromosomes inside of the tetrads separate and move to opposite ends of the cell. The centromeres that hold the sister chromatids together do not split. This will let you know that each new cell will receive only one chromosome from each homologous pair. The cell is beginning to divide. The chromosomes are moving to opposite ends. The centrioles and the spindle fibers are also at opposite ends. The cell is still diploid.
At the beginning of telophase 1, the cell begins the division process. The chromosomes are on opposite sides of the cell, and nuclear membrane begins to reappear around the chromosomes. The cell undergoes division called cytokinesis, where the cytoplasm and cell membrane divide to form 2 new cells. The 2 new cells that are present are now haploid because they do not contain the homologous chromosomes that are in the other cell. Both cells have sister chromatids that still need to divide, however. So both of the new cells will begin another division process, meiosis II, that is similar to mitosis.
Both cells are now haploid, since the original cell is now split into 2 daughter cells. During prophase 2, the centrioles and the spindle fibers duplicate and the nuclear membrane surrounding the chromosomes disappears. The chromosomes can now roam freely about the cell.
During metaphase 2, the 2 cells are haploid. Like in metaphase 1, the chromosomes line up in the center. However, in metaphase 2, the sister chromatids are aligned in the center instead of tetrads. The centrioles and spindle fibers move to opposite ends of the cell, and the spindle fibers attach to the centromeres that hold the sister chromatids together.
In the beginning of anaphase 2, the spindle fibers begin to pull apart the sister chromatids. The centromeres holding the sister chromatids together split, ensuring that each daughter cell will receive each 1 chromosome from each sister chromatid. Both cells are now beginning to divide in late anaphase 2, and the spindle fibers pull the sister chromatids to opposite ends of the cell, and the cell prepares for telophase 2.
Telophase 1 and telophase 2 are very similar. During telophase 2, both haploid cells divide. The nuclear membranes form around the chromosomes in each daughter cell, and each cell undergoes cytokinesis again, forming 4 haploid daughter cells with half the number of chromosomes that the original parent had. So each cell is now a haploid gamete, and meiosis II is over. The complete cell division process of meiosis has now been finished.